An activity neighbourhood analysis looks for significant differences in a property created by small structural changes around a reference compound. These changes are often described as activity cliffs and can represent important structure-activity relationships to guide the further optimization of compounds. To run an activity neighbourhood analysis, select a reference compound in card view and select the Activity Neighbourhood option from the Analyze menu. In this dialog, you can select the number of nearest neighbours to analyze around the reference. By default, these nearest neighbours will be shown as links in card view, and you can also choose to colour the links by the change in the property value. In this case, we will choose the affinity for 5-HT1A expressed as a pKi. You can also choose the colour scale by clicking on the boxes below, but in this case, we will use the default from grey to red. Clicking OK will run the analysis and display the results. In card view, the cards will be displayed as a spiral, with the reference compound in the center and other cards arranged in order of decreasing similarity going outwards. The arrows on the links indicate the direction in which the property value, in this case affinity, increases, and the color indicates the size of the increase. So a short red link indicates a large change in the property caused by a small change in the structure, an activity cliff, while a gray link indicates a small change in property value. For example, here we can see a link indicating that removing a benzyl group from the nitrogen causes a significant increase in potency. But, on the other hand, changing the methoxy group to a hydroxy causes a negligible change. The table summarizes the results for the nearest neighbors, and you can display a heat map highlighting the change in each property between the neighbors. Selecting a row on the table or a bar on the histogram will select the corresponding links in card view. In the next video, we will see how this concept can be generalized to create an activity landscape comparing all of the compounds in a dataset with one another.